Okay, well, thank you, Greg. Well, for those who know me, they know that I love NMR. I have a background in NMR, but I do not have a background in protein NMR or NMR looking at large biological molecules. But being here at Cambridge Isotope Labs for over 20 years, I've gained quite a bit of knowledge from working here, but also from speaking with folks like you that are in the audience. Now, determining the three-dimensional structure of the building blocks of life, such as proteins and nucleic acids, down to the very last atom, is a very tricky business. Even trickier is to determine how these molecules move and bend and bind to other molecules. But the effort is worthwhile because the results, such as three-dimensional structures and binding information and so forth, will be used by medicinal chemists to design new drugs and for other scientists to better understand human health and human disease at the molecular level. Now, although much structural information can be gained by X-ray crystallography and cryo-electron microscopy,
excellent.
It was just my title slide, so you didn't miss anything. So one can use X-ray crystallography or cryo-EM to get structural information, but it is only NMR that is suited to study molecular targets under or near physiological conditions. This is in solution or even in living cells. So using NMR to study protein and nucleic acid requires many things to come together. One needs to have the right hardware. You need a good high field spectrometer, a good magnet, a good probe. You need the right software. You need the pulse programs to make sense of the NMR data and to compute the structures and et cetera. You need the right operator and then right pulse program, you know. So you need to have a lot of things in place to get this work done. But in the end, if you don't have a good sample, all your efforts will be in vain. And nowadays, if your sample is not properly enriched with some set of stable isotopes, you may be in trouble. In fact, you could even say that stable isotopes used in biomolecular NMR is in fact a critical technology. So why are stable isotopes used in biomolecular NMR? The first and foremost reason is they're used to increase signal strength. Now, NMR is awesome. We all know that, but it does have a slight downfall, and it's because it's not inherently super sensitive. And there's many ways to increase the sensitivity. You can have a higher field magnet, you can have a cold probe, or you could just simply turn up the concentration or increase the number of spins. And in the world of biomolecular NMR, that really comes down to increasing the amount of C13 and N15. Large increments, increasements can be made because C13 and N15 normally are at the 1% or below level. And one can easily enrich biomolecules up to 95, 96, 97, 98, even 99%. So one can increase the signal to noise dramatically by enriching the molecules themselves with these stable isotopes. One can also gain signal strength in looking at larger molecules in solution primarily, where the larger molecules tend to rotate slower, allowing other relaxation mechanisms to occur. For example, dipolar relaxation starts to happen when molecules become rather large, you know, around 30 or 40 kilodaltons large, they tumble slower. And their line widths become broader due to dipolar relaxation. And one can fight against that by replacing hydrogens with deuterium in order to reduce that dipolar relaxation. And stable isotopes are used to simplify spectra. As the molecular size increases, the number of resonances also increase, and so does the problem of spectral overlap. Stable isotope labeling can help reduce the complexity of spectra in a few ways. One method is to simply simplify spectra is to use amino acid selective type labeling. And in this type of labeling, one will just add certain amino acids that you'd like to see labeled to an otherwise unlabeled cell culture media. And so your protein will only have residues of which you've chosen to see. Another commonly used method to simplify spectra is to only label the methyl groups of a larger molecule. That is called methyl labeling. We will hear more about that today. Now, with increased signal strength of an enriched biomolecule, one can then use higher dimensional experiments, 2D, 3D, 4D type of NMR experiments to further separate signals in order to overcome signal overlap and to help in the spectral resolution. Now, with this increased signal to noise and being able to simplify spectra in such a ways, one can then conduct experiments to get the information that you really want to, which is elements of structure, the primary sequence, the secondary structure, tertiary, even quaternary structure. And these are gained by various types of NMR experiments. NMR experiments that may look at the internuclear distances between two or more atoms. And this can be done using the nuclear Overhauser effect or NOESY-based experiments. One can use also scalar-based experiments, that is triple label experiments, to look at connectivities, say in the backbone, to walk down the backbone and to look at the various R groups of the amino acids. And one can also use NMR in other ways. For instance, one can look at relative orientation constraints by employing the methods used in residual dipolar coupling or RDC experiments. And it's not only structure. The fantastic about NMR is one can look at dynamics as well. And stable isotopes can help in that area by producing very simple isolated two-spin systems where relaxation can be studied. For example, relaxation dispersion techniques can probe microsecond to millisecond timescales, which are associated with protein folding, for example. And we'll hear some more about dynamics from Lewis K. today. And lastly, one can look at interactions. What's very important or interesting is how a potential drug molecule may bind an enzyme, for example. So one can look at small molecule binding or large molecule binding by having isotopes in the molecules. This is usually done through kind of a procedure called chemical shift perturbation experiments, where one looks at the change of cross peaks as one titrates in a ligand. And one can get the position of the binding as well as the strength and the lifetime of the binding in these types of experiments. And so how does one label a protein? Well, one labels a protein depending on the type of information that you would like to obtain, but also the size of the molecule matters. Very small molecules don't even require any isotopes, You can get a lot of information by just proton-proton homonuclear type of experiments. But now this slide is really dealing with protein. I'll have another slide dealing with nucleic acids, but small proteins, around 10 to 30 kilodaltons. One requires uniform labeling to get structure and dynamic information. And for these size molecules, one will typically use minimal media, and one always has a choice of what type of organism they would like to use to overexpress their protein. And the answer is always E. coli. E. coli is the easiest and least expensive way to go. But in some cases, as the complexity of the molecule increases, E. coli may not have enough or the correct molecular machinery to properly fold the protein. And that is where one will have to go to another organism, like a eukaryotic organism, such as yeast, insect cells, or mammalian cells. And
1 minute,
also,
Kev.
what?
1 minute.
1 minute. Okay.
Yep.
So I talked a lot slower than I had thought. So I'm sorry. So anyway, as the complexity increases, so does the complexity of the types of stable isotopes that are required. One uses deuterium label substrates to label a protein with deuterium in order to look at such things as sparse labeling and to look at the methyl groups. And Lewis K. will talk about methyl labeling as well. On nucleic acids, we label using either NTPs, using the PCR chain reaction, or one can build chains one residue at a time by using deoxyphosphoramidites. We have a couple of new products here. We do have some 2'15F NTPs from Cassia. We get all our nucleotide phosphates from Cassia. They make great products. We also are now reselling some deuterated phospholipids and detergents from FB Reagents. They're located in Bulgaria. These products, there's more on our website. They're perfect for membrane proteins. And we do have 2 aromatic probes, which no one else has. These are brand new products. And they're shown here. There are tyrosine and phenylalanine. So I'd like to thank everybody at this point. And I guess the time is up. Right on the dot. It's right 9:30. So yep. I don't think there's any time for questions, but if you do have questions, you can send a question in the chat, or you can just email me at kmillis@isotope.com. So,
Kevin, we did have one question that you partially addressed. Can you comment on F19 labeling of proteins?
Well, it's using fluorine has been going on for years. It seems to have become more prevalent lately, and it's because fluorine has a very large available chemical shift, and its chemical shift is extremely sensitive to its environment. And one can look at long-range couplings. I think Mei Hong has shown this recently, where one can get quite long-range information using fluorine.
Excellent. Thank you very much. Okay, thanks again, Kevin. Okay,
thanks, Krista. And I'm going to stop sharing my screen so I can introduce our next speaker.